Hi everyone, my name is Dipika Raghuraman and this is my presentation on thalassemia. So thalassemia is a hereditary blood condition and it's most commonly expressed in the autosomal recessive inheritance pattern. So it was first identified in the region surrounding the Mediterranean Sea. So thalassa is Greek for sea and emia is blood condition. So, thalassemia is characterized by errors and defects in the synthesis of the hemoglobin alpha and beta chains, as you can see in this GIF. So these defects can result in hemolytic anemia, among other symptoms. So alpha thalassemia is most commonly expressed in the major and minor forms. So since it's a recessive disease, the major form of alpha thalassemia needs to have two copies of the defective alpha gene. So that it needs to be inherited from both parents. The minor form is, of course, only inherited from one parent, and these carriers don't have many symptoms of thalassemia. Similar to alpha thalassemia, beta thalassemia is also most commonly expressed when there's two copies of the defective beta chain allele. So symptoms of thalassemia, as I mentioned before, thalassemia can cause hemolytic anemia, and other symptoms include jaundice from the defects in the hemoglobin heme breakdown pathway. You get an excess of bilirubin accumulating everywhere in your body. So there you can also see splenomegaly and hepatomegaly. Patients that come in with thalassemia often are presenting with enlarged abdomens from the splenomegaly and hepatomegaly. Major alpha thalassemia most commonly results in stillbirths. The fetus does not survive past birth. And beta major thalassemia, patients having beta can survive up till about middle age, 20s, 30s, 40s. And they often present with Cooley's anemia, and Cooley's anemia is characterized by the jaundice and splenomegaly, but also malformations in the skeletal abnormalities in your face and skull. They commonly need lots of blood transfusions and chelation therapies to keep surviving on a daily basis. So in this picture, you can see on the left are normal red blood cells, and on the right, this is specifically beta thalassemia. You can see the malformations in the red blood cells. So since there are abnormalities in the formation of the hemoglobin, this causes abnormalities in the shape and size of the red blood cells as well. So you can see like the spiky and oval shaped, teardrop shaped red blood cells. And over here you can also see a nucleated red blood cell. This is because since the body is recognizing that there's not many functional red blood cells in the body, since the bone marrow starts pumping out as many red blood cells as possible, and the red blood cells don't have time to denucleate. So you can see in this blood smear, the stain is showing that there's still a nucleus in that red blood cell. So what is the genetic basis of thalassemia? So thalassemia is caused by point mutations. in the mRNA sequence. So these point mutations are generally found in chromosomes 11 and 16, and they are mostly found in the regions that code for the alpha and beta chains of the hemoglobin. So in the HbA and HbB genes. So these point mutations, they can affect the processes such as transcription, RNA splicing, and RNA translation. And they can cause errors in the codon, which affect the formation of the actual hemoglobin protein. So I've chose to focus in on the RNA splicing defect caused by the point mutations. And just as a review of what we learned in class earlier, this is the splicing mechanism. As we learned in class, different SNRPs come in and they recognize the AG end of the first exon and the G end, G beginning of the second exon. And also there's a branch point, an A nucleotide here. So they recognize and slice at the ends of these nucleotides, the AG and the G, and then they form the lariat loop of the intron and then discard it. So there are many errors that can come from this process, especially when you're considering point mutations when they're thrown into the mix. Say that your AG, there's a point mutation that changes the G into a C. The SNRPs won't be able to recognize that site anymore. Instead, they'll be recognizing something over here or here, and it'll cause a mis-lariat splice. So these are the common types of splicing errors you can keep in an intron, or have like a cryptic intron, or like you remove exons. But the most common splicing error that's found in thalassemia is the alternative splice sites. So here you can see that there's missing, so this is the normal exon, and here you can see that there's different sections that are being spliced out because of the point mutations that are happening. You're losing large chunks or keeping in large chunks of the exons and introns. So this can cause different defects in the formation of the alpha and beta hemoglobin chains. So specifically, I'll start with alpha thalassemia. So here you can see in this diagram that the HPA gene, there's defects that can be found. If you have all 4 functional alpha genes, then you're normal. You don't have the effects of thalassemia. If you're missing one, that's fine. You still have the other one to correspond, and it's a silent alpha thalassemia. There aren't any symptoms showing. But if you're losing 2 of your alpha genes, whether it be on different chromosomes, like in the alpha thalassemia found in the African descent people, or if you're missing 2 alpha genes on the same chromosome, heterozygous, as found in the Southeast Asian descent people, it can cause minor symptoms of alpha thalassemia. So these symptoms aren't extremely dangerous yet. But if you are missing most, if not all, of your alpha genes, this can lead to an accumulation of beta chains, since there's not many alpha hemoglobin chains, or there aren't any alpha, to bind with the beta chains. And this can lead to a formation of hemoglobin H, which is formed mostly by beta chain accumulation. Hemoglobin H is very, very poor in oxygen exchange and can lead to hemolytic anemia, which can lead to death, which is why most fetuses don't survive, causing stillborns. So I couldn't find a great picture of beta thalassemia, so I drew my own. So this is a normal red blood cell. As you can see, the hemoglobins have 2 alpha and 2 beta, and they define the shape of the red blood cell. But if you're missing, so if you have the beta thalassemia defect from one parent, and you're missing half of your beta chain genes, then there's an excess of alpha chains, which eventually bind to gamma and delta chains, which can so they're from the fetal hemoglobin and hemoglobin A2 forms of hemoglobin. So they are packed differently, they accumulate and aggregate differently, so they change the shape and size of the red blood cells. As we saw before in the blood smear, the spiky blood cells and the teardrop-shaped blood cells. This is minor beta thalassemia. For major beta thalassemia, you have a complete lack of beta chains. It's a complete beta chain defect. And so no beta chains will form from the RNA translation into proteins. So you have an excess of alpha hemoglobins, hemoglobin chains. So alpha starts to aggregate together, and they can cause disruptions in the cell membrane of RBCs, which can cause hemolytic anemia. So there is no treatment that can cure beta thalassemia or alpha thalassemia. However, there are palliative treatments that can be used to treat patients that have thalassemia. So they can use blood transfusions, like constant blood transfusions over the years, to keep their red blood cell levels up and keep oxygen exchange happening. But when they do that, they have an accumulation of iron, since the heme groups inside the hemoglobin, they carry iron. Accumulation of heavy metals like iron is very dangerous and toxic, so you have to go through chelation therapy to get rid of the irons that are accumulated. Or they can also go through bone marrow transplant, so that the healthy bone marrow from a donor can start creating normal red blood cells in the body. There are a few treatments that are being researched right now. So there's gene therapy and stem cell therapy. In this study that was released just last year, the researchers are analyzing the effect of gene therapy, where they use CD34 cells in order to encourage normal erythropoiesis. And as you can see here in the gray bars, after you started the gene therapy, the red blood cell volume starts to, there's changes in the volume which bring it back to normal red blood cell shape and size. And the number of transfusions starts to decrease as well. So that's the end of my presentation. Thank you for listening.